And nitric oxide also plays a critical role in neural stem cell maturation and integration of these new neurons into existing circuitry. So nitric oxide is a key player in neurogenesis and neuroplasticity. My name is Brendan Vermeyer. I'm a functional medicine practitioner, clinical educator, and researcher, and I'm the clinical education manager for Berkeley Life and the host of The More You Know series. The More You Know is a monthly webcast designed to bring you hot topics and takes on the importance of nitric oxide and blood flow for optimizing the body's natural healing systems. We encourage you to watch, share, and pause the video to take in all of the information. Do you have a topic that you'd like to see us feature? Reach out to us at info@berkeleylife.com. We appreciate you joining us. Now on to the show. Hey everybody, welcome back to the Berkeley Life, The More You Know series. As always, I'm your host, Brendan Vermeyer, and today on this episode, we are going to be discussing the role of nitric oxide in neuroimmune disorders. So this is an area of clinical research and practice that I'm especially passionate about. Neuroimmunology is actually my main area of focus and dare I even say maybe expertise. I'll let you be the judge of that, but I'm very excited to have the opportunity to share some of my knowledge of how nitric oxide is directly implicated in a variety of neuroimmune and neurodegenerative disorders. So be sure to check out some of our past episodes of the More You Know series where we cover a lot of other areas, whether it's cardiometabolic or cognitive and a little bit of everything in between. So as always, of course, I think we have to appreciate the chronic disease epidemic, and the neuroimmune epidemic is on the rise concomitantly. And of course, there's a positive correlation and association here. There's many of the same etiological root cause factors that are driving not only cardiometabolic or chronic disease, but also the prevalent and increasing prevalence of autoimmune disorders and neuroimmune disorders. So we see with 6 in 10 Americans having a chronic disease, 4 in 10 having 2 or more chronic conditions, 1 in 5 Americans are estimated to have an autoimmune disorder, and about 75% of these 50 million Americans affected by an autoimmune condition are women. So we see a much higher rate of women, American women, being affected by autoimmunity than men. And there's certainly a number of theories as to why that is, a lot of estrogen receptor activity theories as well. Now, multiple sclerosis, which is really perhaps the most prevalent of the neuroimmune, neuroautoimmune disorders, is estimated to affect about 1 million Americans. We're seeing this number on the rise. And while some of the other neurodegenerative disorders, such as Parkinson's or Alzheimer's, these are not classically thought of or yet categorized as autoimmune disorders. However, a large and rapidly expanding body of evidence within the field of neuroimmunology or psychoneuroendocrine immunology is very clearly demonstrating that the immune system is directly implicated in Alzheimer's, Parkinson's, and really all of these different neurodegenerative conditions. Now, personally, based on the work that I've done, based on the research I've seen, I do predict that in time, Alzheimer's, Parkinson's, and even things like depression or autism spectrum disorder will be acknowledged as having an autoimmune component. And candidly, the more that we learn about autoimmunity, the more research goes into it, many chronic conditions do have an immunological or autoimmune component to them. So I think as time goes by and research advances, we'll just see more and more of this. And so I'll elucidate some of the implicated mechanisms going on with this to provide some clarity around this. And of course, we see Alzheimer's is one of the leading causes of death, with about 5.8 million Americans having Alzheimer's or related conditions. So certainly, I think we all probably know somebody that might be struggling with a neurodegenerative disorder or a neuroinflammatory disorder. Neuroimmune, like multiple sclerosis, not quite as common, but certainly increasing in prevalence. And I mean, I can certainly think of a number of people that I do know that have either ALS or multiple sclerosis, and even getting diagnosed younger and younger. Now, with the improved technology and testing, of course, our ability as practitioners, clinicians, doctors, healthcare providers to detect these abnormalities earlier is getting better and better. So thank goodness for all the amazing, new biomarkers and new lab tests that we have that we can screen for these things sooner rather than later. So as we say on every episode all the time, because we are certainly the blood flow company at Berkeley Life with our dietary nitrate, nitric oxide products here. So blood flow is always has to be considered the foundation of all wellness and healing. So in this episode, we're going to take that a little bit more specifically. We always discuss the importance of healthy blood flow. We have to be able to effectively deliver red blood cells and oxygen and nutrients, immune cells, transporting waste, hormones, neurotransmitters, short-chain fatty acids, so on and so forth. It all really starts with healthy, adequate blood flow, perfusion, vasodilation. Now, above all else, nitric oxide is, above all else, a vasodilation molecule. It is the molecule that allows the diameter of our vasculature and lymphatic vessels to expand in diameter through vasodilation to allow for more blood flow and lymph flow. So why I'm mentioning lymph, I really mention it on pretty much every episode, because especially with the topic of lymphatic drainage that has picked up a lot of traction in the functional medicine world, well, guess what? The vasodilation of the lymphatic vessels is under the control of nitric oxide. So whether we want to have a conversation about blood flow or lymph flow, nitric oxide is the star of that show. So especially when we're talking about neuroimmune disorders or neurodegenerative disorders or even neuropsychiatric disorders, and we're talking about blood flow to the brain and the parenchyma of the central nervous system, as well as the glymphatic system, which is essentially how we detoxify and mobilize a lot of misfolded proteins and waste products out of the brain through the glymphatic system. Guess what? Nitric oxide is the star of that show. So with that, compromised blood flow, hypoperfusion, hypoxia, ischemia, compromised blood flow can directly exacerbate multiple sclerosis and other neuroimmune disorders. So I want to speak to that a little bit here. We know that the brain is a very energy intense organ that requires a constant supply of blood flow, nutrients, oxygenation, everything else that's being transported in the blood, including immune cell transportation, all day, every day. And so even though the brain is only accounting for about 2% of the body's weight, from a metabolic energy demand perspective, it's accounting for about 20% of our daily energy expenditure, right? It's a very ATP, energy hungry organ with the gray matter. So a few of the relevant mechanisms here that we have to acknowledge and be cognizant of as far as how compromised blood flow can exacerbate and potentiate a neuroimmune disorder such as this demyelinating disorder that is multiple sclerosis is through decreased oxygenation to the brain. If we have decreased blood flow, this will directly cause a more hypoxic or low oxygen tension state within the brain. Now, there's a lot of things that can go on here. Of course, we all can appreciate glucose is the primary energy source for our brain cells. Now, our brain cells can use other energy sources as needed. Of course, if we are very on a low carbohydrate diet, we don't have enough glucose. As we all know, we can certainly undergo metabolic ketosis and produce different ketone bodies in the liver that can act as a secondary fuel source for brain cells, but also glutamate. Glutamate can be produced by astrocytes or macroglia as a backup fuel source under hypoxic conditions. Now, certainly, glucose, ketones are comparable energy sources, but I'm really wanting to highlight if we have compromised blood flow and hypoperfusion to the brain, and we're not getting enough oxygen to our brain cells, to our neurons, there's not enough oxygen to facilitate oxidative phosphorylation and glycolysis. So then what happens is astrocytes or macroglia have to kind of pick up the slack. So under hypoxic conditions, being the key focal point here, astrocytes will create de novo, brand new glutamate. Now, glutamate, as many of us know, is typically thought of as an excitatory neurotransmitter. Now, it's certainly very, very important. It's not a bad thing. But when we have excess glutamate in the brain, it can contribute to anxiety through increased glutamatergic activity. And if that gets too excessive, that can lead to what we call excitotoxicity. Essentially, too much excitatory neurotransmission that can become toxic and damaging to the brain and actually increase neuroinflammation and microglial activation. So under these hypoxic conditions, whether that's due to anemia or compromised blood flow or low nitric oxide or whatever is going on there, in the hypoxic brain, this is going to decrease energy production. It's going to increase glutamatergic activity, which can increase excitotoxicity. Now, certainly the de novo glutamate production by astrocytes under hypoxic conditions, that is a metabolic compensatory mechanism to supply the neurons with glutamate as a backup fuel source. Glutamate can enter the citric acid cycle and be used to create electron donors and facilitate the electron transport chain to produce ATP. But under hypoxic conditions, this can be increased. So we have to restore nitric oxide signaling, vasodilation, blood flow, perfusion, and keep the brain oxygenated at all times. When we have this hypoxic state, it can have defective energy metabolism and cause cellular stress that can increase oxidative and nitrosative stress, as well as inflammation, or rather neuroinflammation. This can also lead to the disruption of the blood-brain barrier. So then, of course, as we have this leaky brain effect, this will allow more immune cells and toxins and pathogen-associated molecular patterns, things like lipopolysaccharide or lipoteichoic acid, or even mycotoxins or spike proteins or whatever it is. It can allow more toxins, toxicants, metals, PAMPs, et cetera, to leak into the brain that is going to drive more glial system or the immune system of the brain, more activation of these glial cells, the macroglia or astrocytes, and the microglia, which is my favorite cell in the body and what all of my work really centers around. And this can also impair waste removal. It can impair the ability to transport waste out of the brain. For example, we have ATP binding cassette transporters that reside within the blood-brain barrier. And these essentially act as like little protein pumps to pump things like amyloid beta out of the brain into peripheral systemic circulation. Under more inflammatory conditions, these ATP binding cassette transporters become impaired. The expression of these ATP binding cassette transporters is downregulated under inflammatory conditions. There's a number of pro-inflammatory cytokines that will decrease the expression of these essentially detoxification waste transporting proteins that transport the waste out of the brain into circulation. Our lymphatic system might also be compromised as well. A major way that we detoxify the brain and remove misfolded proteins that can accumulate in different neurodegenerative conditions. And of course, then also mitochondrial dysfunction can become compromised. So brain energy ATP supply will become compromised. Oxidative and nitrosative species and radicals can increase, things like superoxide or peroxynitrite, for example. And all of this can exacerbate and increase the demyelination and autoimmune process. So we have to be thinking about, how do we restore optimal blood flow in the context of this conversation, specifically to the brain and central nervous system, so we can supply it with the oxygen, the nutrients, the immune cells, the hormones, the metabolites, so that it can properly feed the neurons, the oligodendrocytes, and we're able to remyelinate our nervous system more effectively. Of course, multiple sclerosis specifically is a demyelinating autoimmune disorder. So this is all going to be exacerbated by compromised blood flow, as an example. So this is, of course, where enter the miracle molecule, nitric oxide, which is what we're all about at Berkeley Life. So we have to have adequate nitric oxide for the blood flow, for the vasodilation, the vascular tone, maintaining normotensive blood pressure readings, but also for optimal mitochondrial function, to transmit signals between neurons, to combat pathogens and infections through the activation of inducible nitric oxide synthase expressed by epithelial cells in the intestines, and more so by immune cells such as macrophages and neutrophils and the microglia, which is essentially the macrophage of the central nervous system, different cellular organs, though, as a clinical pearl. So, of course, as I say in every episode, nitric oxide dysregulation is implicated in all chronic disease. I always like to highlight that. And what we're very passionate about at Berkeley Life is equipping practitioners and healthcare providers with the knowledge and the tools to restore proper nitric oxide signaling throughout the body. Now, we do this by leveraging the nitrate pathway. So I cover this in every episode, and I'm going to highlight it again here. Let's look at my more comprehensive diagram. So we'll go through this quickly as we do in every episode. So most practitioners that are familiar with nitric oxide are more familiar with the classic nitric oxide synthase pathway, the NOS pathway. This is our body's innate endogenous way that we can produce nitric oxide through the nitric oxide synthase enzyme. Now, of course, this is encoded in our genes with our NOS genes. We have NOS1, NOS2, and NOS3. NOS1 is neuronal nitric oxide synthase, the isoform of nitric oxide synthase expressed primarily by neurons. And we'll discuss that a little bit further momentarily. NOS2 is our inducible nitric oxide synthase that I was just talking about. So when we have infection or foreign invaders or some sort of immunological activation state, our immune cells will express inducible nitric oxide synthase to create high quantities of nitric oxide to use it as an antimicrobial molecule to fight off the foreign invader or infection. So it's a part of the immunological inflammatory response. Under healthy, controlled physiological conditions, this is a good thing. Under chronic inflammatory conditions, this is not such a good thing. And then finally, we have the endothelial nitric oxide synthase, or eNOS, NOS3. This is probably the most talked about of the NOS isoforms, because of course it's expressed by the endothelium, both in our blood vessels and our lymphatic vessels to regulate vasodilation, right? So we use L-arginine as the nitrogen source, and we require a lot of different cofactors to be able to facilitate the conversion of that L-arginine into nitric oxide. So we need, first and foremost, molecular oxygen. If we don't have enough oxygen, we are not able to facilitate the NOS enzymatic reaction. But we also need a lot of other cofactors, such as bioterin. Some people say biopterin, BH4. So BH4 is a major cofactor. If we have MTHFR or methylation defect or hyperhomocystinemia, our BH4 becomes compromised. If our pentose phosphate pathway is compromised, we have decreased NADPH bioavailability, which is a major redox molecule and cofactor for NOS. We have FMN, FAD, magnesium, heme, iron, glutathione. There are many cofactors that we need to facilitate the NOS enzymatic conversion. So there's a lot of things that can go wrong with the NOS pathway. For one thing, we might have a nitric oxide synthase polymorphism. There are known clinically significant polymorphisms within the NOS1 gene, NOS2, and NOS3, right? So we might have genetic SNPs that predispose us to nitric oxide dysregulation. But then further, if we have a lack of cofactors or a lack of oxygen, or this phenomenon of nitric oxide synthase uncoupling or NOS uncoupling. Essentially, NOS uncoupling is when the nitric oxide synthase enzyme becomes dysregulated or uncoupled. And instead of producing our miracle molecule, the molecule of life, nitric oxide, it's producing superoxide radicals, which can further react with nitric oxide to form an even nastier radical known as peroxynitrite. So with nitric oxide uncoupling, this can be caused by lack of substrate or increased oxidative or nitrosative stress, inflammation, so on and so forth. Now, when we think about chronic disease, which is largely characterized by increased oxidative stress and inflammation, really, I would postulate that anybody struggling with a chronic condition, a chronic inflammatory disease, is likely struggling with some degree of nitric oxide synthase uncoupling. So this is why we don't recommend arginine-based nitric oxide supplements, because if you push L-arginine or cofactors into an uncoupled NOS enzyme, it could possibly just make things worse because you're pumping superoxide instead of nitric oxide. Now, we can largely circumvent the defective NOS pathway by leveraging the nitrate pathway, which, as the research expands, it seems this might be our dominant ability to produce nitric oxide to begin with. It's an ancient pathway that's largely mediated by the microbiome, the oral microbiome that resides in the oral cavity, and the gastrointestinal microbiome. So the literature sometimes describes the nitrate pathway as a non-enzymatic pathway. However, there's a little bit of nuance there and some contradiction in the literature, because some of these microbes, various microbial species and bacterial species that should reside in a healthy oral cavity and a healthy gastrointestinal tract, do express nitrate reductase enzymes. So even though it's facilitated by microbes, it's still an enzymatic conversion of our ability to reduce nitrate into nitrite into nitric oxide. Now, this can happen in a number of ways. The first step, the first site, is the oral microbiota, assuming that we have adequate nitrate-reducing bacterial species within our oral microbiome. So it opens up a whole conversation about oral hygiene. We've talked about oral hygiene extensively on previous episodes of The More You Know, so be sure to go back to some of our earlier episodes where we go deep into the oral microbiome, just cliff notes for today. The second stage, or the second site of nitrate and nitrite reduction into nitric oxide, is the acidic stomach environment. Now, we have to have an adequately acidic stomach environment to reduce nitrate or nitrite into nitric oxide. The third site would be the gastrointestinal microbiome, which should also have nitrate-reducing bacterial species within our gut microbiota, right? Now, there are other sites and pathways, and there's new mechanisms being discovered seemingly constantly of other ways that we can reduce nitrate or nitrite into nitric oxide, such as xanthine oxidase or cytochrome BC1 or complex 3 of the electron transport chain, or aldehyde oxidase. We've also seen that muscle, skeletal muscle, is a major site of nitrate storage. So our goal at Berkeley is we want to maintain a healthy level of nitrate circulating in the body. We can think of this as a sort of nitrate pool or nitrate reservoir, similar to an amino acid pool, right? We have to have adequate circulating amino acids so we can make proteins and enzymes and build lean mass and all the things. It's similar conceptually with our nitrate reservoir. We can store nitrate in muscles, we can circulate nitrate throughout the body, and we concentrate a high degree of storage of nitrate, 10 times the concentration of the plasma in the salivary gland. This is why our salivary nitrite test strips are a really great way to assess whole body nitrate, nitrite, and nitric oxide by testing the saliva for nitrate levels, because what's going on there is we store and concentrate a lot of nitrate in the salivary glands. And so then when we have a salivary secretion, that hopefully nitrate-rich saliva is going to interact with that oral microbiota, and assuming that we have adequate nitrate-reducing bacterial species in our oral microbiota, we should be able to immediately reduce some of that nitrate into nitrite, which is what the test strips are detecting through a Grice reaction that detects the nitrite ions in the saliva. That's how these test strips really work. And it's a very versatile clinical tool that we can use. So with that, now, of course, whether we're making nitric oxide through the NOS pathway or the nitrate pathway, nitric oxide only has a half-life of about 3 milliseconds in the blood. So it's very quickly going to become oxidized back into nitrite or nitrate. You can very much think of nitrate and nitrite as the storage form of nitric oxide, and we will reduce the nitrate and nitrite as needed in a tissue-specific manner, right? So with that, of course, as we age, we naturally lose a lot of our endothelial function. It's estimated by the time you hit 40, you're at about 50% capacity to produce nitric oxide in your endothelium. Now, as I say every episode, and I said before, it's not just a matter of aging or senescence or inflammaging, but also just chronic disease in general, because now we're seeing chronic inflammatory conditions in younger and younger individuals. So it's not just about aging or individuals over the age of 40. In my personal and professional opinion, the only people that would really benefit from an arginine-based nitric oxide supplement are people that are younger, ideally under the age of about 30, and metabolically fit and healthy. And unfortunately, that's just not the composition of the American population. So this is why we should really be leveraging the nitrate pathway and using dietary nitrate to boost and restore nitric oxide functionality throughout the body, right? So of course, with nitrate, you know, in a perfect world, we would be getting most of our nitrate from the diet. Where do we get dietary nitrate? Our dark green leafy things, primarily our spinach and arugula and bok choy. Beets, of course, beets have become a very popular nitric oxide blood flow supplement because beets do have, on average, a higher concentration of dietary nitrate, or NO3. Now, of course, as we can all appreciate, first off, how many of our clients and patients are actually eating enough dark green leafy things? Speaking from experience, not very many, right? So we have to ensure they're getting a therapeutic dose of nitrate every day, whether they're getting it from a spinach salad with beets or a green juice, or our dietary nitric oxide foundation product. Or also we have our dietary nitrate prebiotic gum, which is designed to store the oral microbiota. And we'll talk about that a little bit more momentarily. Now, because of soil quality, because of farming practices, because of region where the produce was grown, nitrate concentrations in produce can, of course, fluctuate. As we always like to say in this industry, let's test, not guess. We don't need to guess to see if they're consuming enough nitrate. We can test using the salivary nitrite test strips, reflective of their nitrate intake, circulation, and the ability to reduce that nitrate through the oral microbiota. So getting back to the neuroimmune conversation, to continue some of that, now here's a number of mechanisms by which nitric oxide can be very, very helpful and supportive for neuroimmune health. Of course, increasing blood flow and oxygenation, as we comprehensively discussed a few minutes ago, but also helping support healthy neurotransmitter signaling, neuroendocrine signaling, mitochondrial function in the brain and central nervous system, regulating inflammation and oxidative stress. And nitric oxide also plays a critical role in neural stem cell maturation and integration of these new neurons into existing circuitry. So nitric oxide is a key player in neurogenesis and neuroplasticity, which is how we are able to develop memories, learn new skills, rewire the brain, rewire our belief systems, repair the brain, so on and so forth. Of course, also nitric oxide is critical for regulating the tight junctions of the blood-brain barrier, so we have an intact, healthy blood-brain barrier that is not allowing any noxious elements to leak into the brain from the periphery. Of course, the endothelial function, the blood-brain barrier, of course, is endothelial cells and a blood vessel in itself, and promoting healthy neuroimmune activity. Like I said before, if our oligodendrocytes are not getting enough oxygen, they can't effectively remyelinate our neurons. So when it comes to neurotransmission, which certainly is implicated in neuroimmune disorders, neurodegenerative disorders, and of course, neuropsychiatric disorders as well, nitric oxide actually acts as a retrograde gasotransmitter. So this is really, really cool. Retrograde as in it moves backwards, right? So when we have neurotransmitter activity, the presynaptic neuron will release various neurotransmitters from stored vesicles and sort of dump that neurotransmitter into the synaptic cleft, where it will translocate across the synaptic cleft and bind onto its designated receptors on the postsynaptic neuron, right? Now that postsynaptic neuron will then actually express neuronal nitric oxide synthase, and that nitric oxide gas will diffuse backwards, retrograde across the synaptic cleft and modulate the release of those neurotransmitters from the presynaptic neuron. So the punchline, the take home here is nitric oxide has a very profound effect on regulating neurotransmitter release and activity at the synaptic level. Huge, right? Now, nitric oxide, as I just mentioned, actually plays a critical role in helping neural stem cells mature and develop and grow up into fully functioning neurons. And it also is related to how these new neurons then get integrated into our existing neural circuitry. Neuroplasticity, neurogenesis is how we repair, rewire, and protect the brain. We have to have adequate neurotrophic signaling to maintain a healthy brain throughout life and to delay neurodegeneration, right? So, of course, the blood-brain barrier aspect that we just talked about, nitric oxide has a regulatory effect on those tight junctions, the claudin proteins that help to hold the brain together. We see that, things like homocysteine can bind on to NMDA receptors on the blood-brain barrier and activates an intracellular pathway that then decreases the expression of claudin 5 that can hold these tight junction proteins together. We have to have adequate nitric oxide signaling to maintain these tight junctions so we don't develop that leaky brain that can then drive a lot of neuroinflammation. So that takes me to just a quick little 101 on the subject that my whole career centers around, which is microglial activation. And we don't need to go super in depth for today. But again, those microglial cells that make up about 15% of the brain's cell population. So they're sort of small and few, but very mighty, right? So under normal physiological conditions, we have these resting or quiescent microglial cells, and they're really just patrolling. They're moving around your brain. They're patrolling the environment, surveying the milieu and the microenvironment to make sure that there's no foreign invaders or any inflammatory activity going on. So they're there to basically be the sentinels and the guardians of the central nervous system. Now, certainly microglial cells are directly implicated in neurodevelopment and neuroprotection throughout life. They're both the guardians and the architects of the central nervous system, which is why I am so obsessed with them. Now, depending on the cytokine, chemokine, ROS milieu or microenvironment, when these microglial cells receive input signals from the environment that there's a foreign invader or some sort of inflammatory stimuli, lipopolysaccharide, interleukin-6, TNF-alpha, interferon gamma, so on and so forth, any sort of inflammatory signal, this is going to activate the microglial cells and cause them to shift their morphology and polarize into more of an M1 dominant phenotype. The M1 phenotype is pro-inflammatory. It's cytotoxic. Its job is to fight off foreign invaders, to contribute to the neuroinflammatory response, to serve a protective mechanism. Now, again, under normal regulated physiological immunological conditions, this is a good thing. The problem is when we have excessively M1 activated microglia for too long. And this is antithetical in contrast to the anti-inflammatory neuroprotective M2 phenotype. So we can't blame the microglial cells for being dysfunctional. The glial cells are simply responding to the milieu, to the environment, to the microenvironment that they're bathing in, that they're marinating in. So the problem with most Americans, because of chronic disease and chronic inflammation, is most Americans have too much M1 microglial activity in the central nervous system, as well as too much M1 macrophage polarization throughout the body, throughout the tissues, throughout the periphery. So this drives too much inflammation chronically that can obviously cause tissue damage and degeneration. In this case, we're talking about the brain and the central nervous system, right? So the microglial cells are directly involved in all neurodegenerative conditions and neuroimmune disorders. They are the stars of that show. So all of my work in my career is teaching practitioners how do we modulate the input signals that the microglial cells are receiving to harness the healing power of the M2 phenotype and ultimately have resolution to the neuroinflammatory process. The problem is neuroinflammation is a fire that keeps itself going. It's a self-perpetuating fire. As we have more M1 microglial activation, this is going to potentially cause more damage to the environment, more collateral damage. So as we have more of these damage-associated molecular patterns that are sort of contaminating the parenchyma of the central nervous system, these DAMPs will bind on to pattern recognition receptors on the microglial cells that then just further activates the M1 pro-inflammatory phenotype. So to summarize all of this, I know that for those of you that maybe this is new information, it can be a little bit complex. Some of you are already familiar with this information with microglial activation and polarization or macrophage, so on and so forth. But from a neuroimmune, neuroprotective overview, at a very high level, and if we simplify this, I would argue that the secret to neuroprotection and remyelination in multiple sclerosis, or to decrease the rate of degeneration, we really have to shift the glial cells away from this excessive M1 phenotype to more of a neuroprotective, neuroregenerative M2 phenotype. So a lot of this, and of course then, well, Brendan, how do you do that? That gets into, that's a big question that goes a little bit beyond the scope of this conversation, because it could be a lot of different root cause factors that are driving the M1 activation. But in regards to nitric oxide, I would argue that the first thing we need to do is optimize blood flow and oxygenation and nutrient delivery, because ultimately, if we want to shift the morphology and the phenotypical expression of the glial cells, we have to change the environment. We have to change the milieu. We have to modulate the cytokine, chemokine, ROS microenvironment that these microglial cells are hanging out in, right? And so to do that, we have to promote healthy blood flow and nitric oxide signaling. So we have adequate oxygen and nutrients, immune cell transport, waste transport and detoxification, healthy lymphatic activity as well. This is how we can correct the environment to then change the activity of the glial cells within that environment to promote more of this anti-inflammatory neuroprotective M2 phenotype. Now, of course, pharma is creating lots of new drugs and monoclonal antibodies that can, block or neutralize different inflammatory stimuli. So it depends on what root cause factors might be contributing. But in relation to nitric oxide, it's all about the blood flow, oxygenation, nutrient delivery, so on and so forth, right? Maybe if we had adequate nitric oxide signaling, we would have a more intact, blood-brain barrier, more mitochondrial functionality, so on and so forth. So then we don't have leaky brain. So then we don't have more pro-inflammatory signaling that's driving more M1 activation. So it can get really complex really quick as we factor in all the different root cause factors, but nitric oxide restoration and healthy nitric oxide signaling can be a very critical component here. A lot of times in more inflammatory or neuroinflammatory conditions, we might have eNOS uncoupling or nNOS uncoupling with upregulated iNOS activity, because these glial cells, the microglia, they do express iNOS. So the microglia, when activated, might be producing high levels of nitric oxide or superoxide or peroxynitrite as part of the neuroinflammatory response. But what we want is to decrease the inflammatory load in the brain and shift over to this M2 phenotype. So part of it is we have to get the eNOS, such as in the blood-brain barrier, back online. We have to get the nNOS back online and recoupled and try to quiet down this neuroinflammatory microglial response. This is a lot of information packed into, a pretty short lecture here. So this is a graphic that I made previously that illustrates this really, really well. And what I like about this graphic that I made is we have the blood-brain barrier that expresses the eNOS, we have the neurons that express the nNOS, and then we have the microglia that express the iNOS. So there's actually a lot of nitric oxide activity going on both in the normal physiological condition brain and the neuroinflamed brain, but different activities, different isoforms being either uncoupled or upregulated. So we want to restore homeostasis to this nitric oxide system within the parenchyma of the central nervous system. Because if we can't effectively nourish and feed the oligodendrocytes, they can't effectively remyelinate our neurons in our nervous tissue. And of course, then we have more demyelination, more neuroinflammation, and more neurodegeneration, right? So again, it really starts with healthy blood flow and nitric oxide signaling at a very foundational level, right? Now, with this, a little bit of a summary slide here that encapsulates some of the mechanisms and pathways that we've been discussing throughout this short lecture. So things to be looking for in your neuroimmune patients, of course, blood pressure. What a basic and fundamental metric of human health in general, and what nitric oxide is really best known for. And of course, at Berkeley, we have our gold standard clinical trial that showed a 12.5 systolic point reduction in blood pressure using our nitric oxide foundation product with no other changes. So also be looking out for extremity peripheral neuropathy or cold hands and feet, indicators of poor circulation or hypoperfusion, fatigue, cognitive deficits, and of course, especially with more progressed neuroimmune activity or neurodegeneration, there might be more cognitive decline, more symptoms of dementia, executive function deficit, and even different neurological symptoms, whether it's motor movement impairment or the neuropathy, but it could also present metabolically with weight gain or weight loss resistance, sexual function issues, blood sugar issues, so on and so forth. So many different things to be considering. So by supporting optimal nitric oxide, we can allow everything else to work better. So this is why we have our total nitric oxide solution system that starts with our salivary nitrite test strips. And then, of course, we really want this threshold pink level on the test strips pretty much all day, every day. That's really the goal in my mind. And so we do this by building a foundation with that nitrate reservoir using the dietary nitrate capsules, our nitric oxide foundation product. Now, if they have a deficient or dysbiotic oral microbiota, we can actually use the prebiotic effects of the dietary nitrate in a gum matrix. So that way we're allowing that oral microbiota to bathe and marinate and feed on the nitrate supplied in that gum matrix, right? So it's a therapeutic gum to supply that oral microbiota with essentially the food, the prebiotic, the substrate to allow those nitrate reducing bacterial species to flourish in the oral cavity. So we have better nitrate, nitrite, nitric oxide conversion in the oral cavity. So there's a rapidly growing body of evidence and research that demonstrates how oral hygiene directly impacts metabolic health, whole body health, neurological, neurodegenerative, neuroimmune conditions, so on and so forth, right? So even just thinking about the role of oral hygiene practices, making sure that the oral microbiome is intact and effectively converting the nitrate to nitrite. And that's what these test strips are reflective of. So if somebody is already consuming a lot of dietary nitrate from their diet and or supplementing with the nitric oxide dietary nitrate foundation products, if they're not effectively converting the nitrate into nitrite, they might be getting relatively white test strips. They're not converting on the test strip, even though they're ingesting sufficient nitrate. So what's going on there? That's really implying that they probably have an oral microbiota deficit or deficiency, or at least a deficiency of the nitrate reducing bacterial species. And this is exactly what we created the prebiotic nitrate gum for. Now, especially to wrap up this neuroimmune conversation, we have our cognitive support, which I feel gets overlooked too much. This is a really great cognitive support product, and it's not just good for cognition because the three botanicals that make up this formulation with the Alpinia galanga, the lutein, and the broccoli sprout, they have a lot of antioxidative and anti-inflammatory properties. Now, we at Berkeley have technically not done our own research to substantiate this. However, if you go digging into the scientific literature, you can find papers that demonstrate that all three of these botanical ingredients can exert these anti-inflammatory effects and even possibly have a down-regulating effect on the inducible nitric oxide synthase. So actually, this cognitive support as a clinical pearl can be very complementary to our nitric oxide system because it can help to combat the inflammation oxidative stress that might be driving more NOS uncoupling, or it can also help to qualm the neuroinflammatory or inflammatory response with that iNOS isoform. So it's a very complementary formulation to help protect the nitric oxide molecule. And then the nitrate can also help to restore nitric oxide synthase coupling. So it's actually a very eloquent system to restore healthy nitric oxide signaling in the body, the brain, and the central nervous system. So this is the More You Know series, the Neuroimmune edition. I hope you've really enjoyed this episode. It's certainly my favorite subject with neuroimmunology. So I really hope that this empowers some of you practitioners and clinicians that might be working with neuropsychiatric patients or neuroimmune patients, neurodegenerative patients, and helping them restore their nitric oxide system to help them feel better faster. So that's the More You Know. I'm Brendan Vermeyer. Thanks for tuning in today. Remember, health goes where blood flows. Follow Berkeley Life for better blood flow tips, science, and education.